Hey, Mommy.
Huh?
You know what? We watched a video in our biology class, and the video is wrong.
What? The school is teaching you wrong things?
Well, not exactly wrong, actually. It's just oversimplified.
What's it about?
It's about cell division.
Okay.
So, in the video, it says that when a cell is about to divide, the genes in the nucleus are replicated and split equally into the two new cells.
Isn't that right?
Well, it's oversimplified. The real process is much more complicated.
All right.
So, basically, you know that cellular division is also known as mitosis, right?
Yeah, you talked about that last time.
Yeah. And let me share with you some of the parts during mitosis, okay?
Okay.
So, first, when a cell is about to undergo mitosis, the genetic material, which is the DNA in the nucleus, they're not only replicated, they condense. They condense into 46 pairs of chromosomes.
All right.
Originally, there are 23 pairs, but after replication, there are 46 pairs, right?
All right. Yeah.
Yeah. And at the meantime, the centrosome, which is sort of like the control center for microtubules. Microtubules are like little strands of proteins used for stabilizing cellular structures. So the centrosome splits into two parts too, and the two new centrosomes move towards the two poles of the cell.
Mm-hmm.
And after these two steps are finished, the nuclear envelope rips apart, exposing the chromosomes to the cytoplasm. And
What?
cytoplasm,
What is
which
that?
is the fluid inside our cells.
All right. Okay.
And while the nuclear envelope rips apart, new microtubules will radiate out from the two centrosomes and reach towards the chromosomes until every one of them touches a chromatid. Well, some of them touch each other to push the cell apart. After all the chromosomes are lined up on an imaginary plate, sister chromatid adhesion would be broken. That means that the chromatids are separated, which means that once each pair has 4 chromatids, and now, after splitting apart, each pair only has 2 chromatids, and they move towards the two poles of the cell. Well, it's not that the microtubules pull the chromosomes in, it's the chromosomes moving along the microtubules with the help of motor proteins and breaking it up at the meantime. Then, after all the chromosomes are condensed at the two ends of the long cell, cytokinesis starts. Cytokinesis is the process in which the cytoplasm would be divided equally into the two new cells, and the plasma membrane in the middle of the long cell now begins to get more and more narrow until
it
the
breaks.
one cell breaks into two cells.
Right.
And then in the two individual new cells, a nuclear envelope reforms around the chromosomes, and the chromosomes turn back into chromatid. Yeah, that's the whole
Wow.
process.
Well, sounds really complicated.
Yeah, it's really complicated. The video that we watched in our biology class is really oversimplified.
I'm so glad that you shared with us the right one.
Well, you're welcome.